I'm Theresa Deisher, the founder and chief scientific officer of AVM Biotechnology. I obtained my PhD in molecular and cellular physiology from Stanford Medical School, and I did my postdoctoral fellowship training in hematology at the University of Washington. Since that time, I've spent my career in the pharmaceutical industry, working with companies like Repligen, Zymogenetics, Immunex, and Amgen. And in my career, I had the opportunity to do everything from the most basic benchtop discovery research all the way to leading clinical development programs. And my experience at these really outstanding pharmaceutical companies prepared me to found and to lead AVM and to get us to where we are now. So I'm a very data-driven person, and that goes back to my graduate research at Stanford University, where I worked on beta blockers for heart failure. And at the time, this was in 1985 to 1990, and even as late as 2001, the experts in the field said that beta blockers would kill heart failure patients. However, there was good data that said otherwise. And so we conducted the preclinical research and then ultimately the clinical research to prove that beta blockers were of great benefit to patients with heart failure. And they were ultimately approved, and within a year had become the gold standard for heart failure patients. So throughout my career, I've found myself oftentimes maybe going against the flow because the data said differently. That included the discovery of the first adult pluripotent stem cell that I made while I was at Zymogenetics. Discovered that cell and actually patented it. And here at AVM, we're also very data-driven. And our work has identified a novel, absolutely fascinating immune cell that is by nature bispecific. Now, if people don't know what that means, there are all kinds of companies out there genetically modifying cells, trying to make them bispecific. And what that means is that they can target two signals. The cell that our drug induces and mobilizes is naturally bispecific. And so that is really responsible for the preclinical as well as the clinical anti-tumor activity that we've observed. So we've definitely had several eureka moments during AVM's research and development. And what happened as we elevated the dose in the mice doing our preclinical modeling and research was that a novel immune cell became evident on flow cytometry. And flow cytometry is just a way that we scientists use to be able to see cells. And we can see what types of proteins these cells express. And a very novel, absolutely fascinating immune cell was induced at high doses of our small molecule in the mice. And it's also induced in our human patients. It's a very fascinating cell that expresses two proteins that drive it home to and kill cancer. Cancer treatments themselves often cause other diseases. For instance, chemotherapy causes secondary malignancies. Some of the newer immunomodulatory drugs cause secondary malignancies. That means the treatment for cancer causes another type of cancer to develop. And by reducing the exposure to those other drugs, we reduce those risks and make short-term as well as long-term quality of life better for patients. We have really exciting preclinical data demonstrating that we do indeed do that. We've had some pretty exciting responses. We treated a 39-year-old woman who had a central nervous system tumor. That's a brain tumor. It was in the right side of her brain, very large. And this woman was actually in a nursing home because her symptoms and her clinical deterioration was so bad. She was a young mother, 39-year-old young mother, and she had failed previous treatments, chemotherapy and other treatments. So we enrolled her in our clinical trial, and 24 hours after she was dosed, she began making white blood cells, and she has a complete remission of her brain tumor, her brain lymphoma. So very exciting for us. We had another patient, also a young patient, a 40-year-old patient who also had a brain tumor, who was treated with our drug and is in complete remission. So those are just two of the patients. We're taking all types of non-Hodgkin's lymphoma. So those were two diffuse large B-cell lymphoma patients. We've had a peripheral T-cell lymphoma patient who had a very, very poor prognosis. All of our patients are heavily pretreated. It means they've failed everything that they could possibly try. And that patient actually had a very rapid response and remission and a long-term remission. She was disease-free completely for 8.5 months. Her health improved. She was actually able to tolerate additional treatments later, and she is still alive 16 months later and with stable disease. So very, very promising and exciting responses. It's one thing that gets me up every morning, is providing hope to patients who have no options. And I mentioned that young mother who didn't make white blood cells, and so who couldn't qualify for anything, who started making blood cells, and then her brain tumor was also completely eliminated. In all of our patients, regardless of the degree of response, because not all of our patients have had their tumor completely eliminated, but they all have a clinical benefit and health improvements. In compassionate use, treated a 77-year-old imminently terminal adenocarcinoma patient who had throat carcinoma. And he sent an email and said, I walked through a park, an entire walk that I wasn't able to do before your drug. We have a 60-year-old patient who did not qualify for our clinical trial. And there are many reasons why patients can or cannot qualify, but clinical trials have to be very controlled, right? And they have to meet certain inclusion criteria, but he was too sick to be enrolled on the trial. And so we have treated this gentleman under compassionate use. He had lymphoma throughout his body, so that's called lymphadenopathy, and also disease almost systemically in his bones. And after the first infusion of AVM 0703, the lymph node, the lymphadenopathy, was completely cured, and the bone cancer was slightly reduced. And he's been repeat dosed and has had deepening responses. So more and more of the bone cancer eliminated with each dose. And we received an email from the clinical research nurse who had seen this patient at a restaurant and just told her how wonderful he was doing and asked her to thank the sponsor for giving him back his life. Our focus is on completing our phase 2 expansion cohorts in relapsed refractory non-Hodgkin's lymphoma so that we can apply for marketing approval. We are interested in additional cancers, and we've submitted a grant application to the National Cancer Institute to collaborate with the Pancreatic Cancer Action Network. So our FDA approved open and enrolling clinical trial. It's a special kind of trial. It's called a adaptive design clinical trial. And what we do is, since this drug in these doses has never been used in humans, is that we do a safety portion, and we start at a low dose and we slowly work up to higher doses, and we evaluate the patients predominantly for safety. We have activity in the hypoxic pancreas, and we have activity against cold tumors means that we may really be able to improve outcome in these patients where the prognosis is just so dismal. We really see AVM as part of the first or second line treatment for patients. And ultimately the potential to be part of the treatment regimen for all types of cancers, where we would reduce toxicities to the patients. We have an open IND, an investigational new drug application. And in our application to the FDA, we said we want to do this phase 1 dose escalation and then immediately move into phase 2 without requiring additional approval, right? If we meet these criteria, and they're strictly laid out, and it's related to safety, and the safety review committee actually determines that. They meet and they discuss the safety and whether we should move to a higher dose. We have approval to move straight into phase 2, which we're doing, starting out with a new dosage, completely new dosage, right? We had to establish safety, and we established safety for 1 dose. But in our compassionate use patients, they're actually allowed to be retreated, and we haven't seen any evidence of accumulating toxicities, and we have deepening responses in these patients. So what we'll be doing is we'll be treating patients in cohorts or buckets. And we need at least 18 patients in each bucket to be able to obtain our pre-specified target response. Well, there's a program called accelerated approval. And if we continue to see the responses that we're seeing now, we would anticipate that we'll need 18 patients treated in a specific dosing regimen, or potentially 18 patients with a specific disease subtype, right? And our statistical experts and biostatisticians have done all of the modeling and everything, and came up with the number that we need 18 patients. And our goal is to show that we have a response in 30% or more of the patients. And right now, our response rate is much higher than that. We have patent protection or intellectual property. And I have a lot of experience with patents working at Zymogenetics. I worked with the patent attorneys to file over 64 patent applications. And those patents actually, besides bringing in significant revenue to Zymogenetics, were very strong enforceable patents. We have a very strong, very unique patent writing strategy that gives us very strong patents. And we have had third-party challenges that we readily overcame. Everyone here is really driven by that, the patients. And so when we get an email from a patient thanking us for giving him back his life, or a young girl that we've treated in New Zealand who had a tumor, abdominal tumors, huge, so rare, there's actually not a standard of care for her, who has had just an incredible response and continues to be treated. And we get emails from that mother. We work very long, very hard hours, and that makes it all worthwhile. For me, it's also personal. My father was one of our compassionate use patients. He was diagnosed with an inoperable and chemotherapy ineligible brain squamous cell carcinoma that was just gruesome. And we were able to treat him under compassionate use. And he had an amazing response. I was there when he was treated, and I was working. The infusion was not even over when the clinical research nurse called me over and said, "You have got to see this." I had no idea this would be so rapid, but his tumor was growing out of his eye, and you could already see it receding. It was an amazing response. I'd like to say that we cure every patient we've treated, but we haven't. But when patients respond, and a significant number of our patients respond, it is amazing. It's an absolutely amazing response. So that's what, drives me to do this work. And I think that that's what drives everyone who works here. Many people know that I lost my younger son, Henry, to Burkitt's lymphoma when he was 14 years old. Unfortunately, we did not have AVM 0703 at that time, or he might be here with us today. The 0703, when you're developing a new drug, you're told companies give it initials, AVM, Amgen calls things AMG, number. And so when we were told to choose a number, I chose 0703 because that's the day that Henry died, July 3rd. And to me, it just is an auspicious date for a young man who would want everybody else to have hope. So people think that I'm developing this drug because my son died from Burkitt's lymphoma, but it's actually quite the opposite. We were always interested in this drug for Burkitt's lymphoma because Burkitt's lymphoma attacks and grows in the germinal centers in your body. And we knew back then, we didn't have clinical stage material, we just had research material, that we could have a just incredible impact on the germinal centers and the lymphomas. So the irony is that my own son was diagnosed with Burkitt's lymphoma. So that's not why we're here, or why we do what we do, but it certainly drives me to push, to persevere. Startups can be very difficult, particularly women-founded startups. I don't want another mother or father to hold their dying child if there's hope that we can provide. So that definitely keeps me going. The novel mechanism of action, the novel immune cell that our drug induces, the clinical benefit it provides, and that I mentioned sort of immune reset. So people's immune systems get exhausted with age and disease, and we get them sleep so they recover, basically, sort of turn that clock back. So it's very cutting edge, it's very avant-garde, and we've gotten top experts in the fields as our advisors, Dr. Frank Butgereit, Dr. John Pietro Dotti, experts in immunology and experts in these cell therapies. I'm also invited by the National Cancer Institute to serve on special grant review panels now, where they're looking at applications for very disruptive, innovative technologies. There were eureka moments that's very exciting, learning about a new cell, understanding the biology, while we're driving the clinical trial forward, and we're gaining the support of so many outside experts who work with us on our publications, on our plans forward. Anytime is an exciting time to join AVM Biotechnology, but I would say particularly at this time. It's so exciting